Let's get started. Thank you everybody for joining. This is our 9th webinar in our Cure 360 webinar series. And in case you've missed some of the other webinars, please go to our website, hit the YouTube channel link, and then we have a playlist called Cure 360 webinar series, and you'll be able to see recordings for any one that you've missed. So before we start, I thought it would be helpful to give a little bit of context to what we're going to be discussing today, which is MECP2 reactivation. And that is one approach of 6 approaches that we're pursuing that you can see here on the screen. So gene replacement, gene editing, MECP2 reactivation, RNA editing, RNA transplicing, and protein replacement. And I'm joined today by 3 speakers, Sanchita Bhatnagar, Kathrin Meyer, and Bryn Martin. Sanchita Bhatnagar is an assistant professor at the University of Virginia in the Biochemistry and Molecular Genetics Department. She's a scientific advisor to Alcyone Therapeutics. And her research focus is gene expression regulation and how that regulation might be exploited to develop therapeutics for neurodevelopmental disorders and also breast cancer. I met Dr. Bhatnagar, gosh, it was almost 10 years ago when she was a postdoc in Michael Green's lab at the University of Massachusetts. And we were funding the lab to work on MECP2 reactivation strategies. She has since moved on and started her own lab, but we're so pleased that she stuck with it and is still working on Rett syndrome. Kathrin Meyer is a principal investigator at Nationwide Children's Hospital. She's also an assistant professor at Ohio State University, and she's the chief scientific advisor to Alcyone Therapeutics. And her research focus is the development of gene therapy for neurological diseases. I met Dr. Meyer probably also around 10 years ago. She was working in the lab of Brian Kaspar, who was one of our lab members and part of our gene therapy consortium. And when he moved on to industry, she stayed and took over his lab. And again, I'm so thrilled that you stayed with Rett syndrome. And then we're also joined by Bryn Martin. He's Vice President of Research, Precision Delivery, and Cerebrospinal Fluid Sciences at Alcyone Therapeutics. That's a real specialty, which I had never even heard of before, Bryn, so we're just delighted to have you join us. He's also an adjunct associate professor at the University of Idaho. And so Dr. Bhatnagar is going to get us started. She's going to be discussing microRNA sponge technology and how that's going to work to reactivate the X chromosome. Dr. Meyer is then going to take over, and she's going to talk about how she's going to package the microRNA sponge into an AAV vector. And then we're going to end with Dr. Martin, who's going to talk about Alcyone's proprietary technology that allows us hopefully to deliver that AAV vector broadly throughout the brain. Please put your questions into the Q&A and we'll address those questions at the end. So I'm going to stop sharing and Dr. Bhatnagar is going to take over. You're muted.
Okay.
There you go.
Now?
Yes.
Okay, great.
And we can see the presentation.
All right. So thank you very much, Monica and the team, for inviting us to talk about our gene therapy for Rett syndrome based on X-Reactivation technology. It's a pleasure and honor to be talking to the community of patients and parents of Rett syndrome. So thank you very much. As Monica set the stage up, I'll be taking a lead and talking about the X-Reactivation based approach. And then I'll pass the baton to an expert in gene therapy field, Kathrin Meyer, and then my colleague, Bryn Martin, will be talking about Alcyone's approach of delivering this in vivo. So I would like to begin by talking about MECP2, which we all know is a key genetic driver of Rett syndrome. Over the years, we have come to realize that MECP2 is an important protein for neuronal maturation, as well as several housekeeping functions that include cell signaling, transcription, protein synthesis, and chromatin organization. Over the last decade or so, we tend to believe that most neurodevelopmental disorders can be reversed. At least in preclinical studies on mice show that expression of a gene that is defective in these disorders can reverse the neurological and morphological features. It is also true for Rett syndrome, which has been elegantly demonstrated in the MECP2 knockout mice. And this idea has led to several therapeutic approaches, which Monica talked about in the beginning, that include expression of MECP2 protein using gene therapy, either the full version or the truncated version, RNA editing, RNA replacement, gene replacement, and also X-reactivation based approach. This approach was, as Monica said, was first at an early stage. I started working on when I was in Dr. Michael Green's lab as a postdoc, and was supported by Rett Syndrome Research Trust as well. But this is a perfect point to also acknowledge the contribution of Rett Syndrome Research Trust, where they have been involved in most of these approaches from the very beginning. And I truly believe that contribution and efforts of Rett Syndrome Research Trust has enabled us to be where we are with all these programs. So I thank you all for being such a great supporter of research for Rett syndrome. So as I said, what I look for is what my lab studies is X chromosome inactivation. What is X chromosome inactivation? Well, all female cells have 2 copies of X chromosome. One that they inherit from mom and the other from dad. To balance out the gene expression between the male and the female cell chooses to inactivate 1 copy of X chromosome. Now this choice is random, and it so happens that half of the cell choose to inactivate dad's X chromosome and express the genes that are present on the mom's X chromosome. And the other half express gene from the dad's X chromosome and inactivate mom's X chromosome. As a result of this X chromosome inactivation phenomena, all females are mosaic for the X-linked gene expression. And this is also true for MECP2 deficiency in Rett girls. Half of the cells are expressing the healthy copy of MECP2, whereas the other half are expressing the inactive or the defective copy of MECP2. We believe that this sick cell, even though it's defective because of the lack of MECP2, holds the key for its own cure because they carry a healthy copy of MECP2 gene on the inactive X chromosome. So if we can express this healthy copy, we can compensate for the deficiency of this gene in the sick cell. And that's exactly what our approach is. It is based on reactivating the X chromosome to express healthy copy of MECP2 in these defective Rett cells. There are important considerations to use this approach as a therapeutic for Rett syndrome treatment. And one of them is the optimal expression of MECP2 in the cells. We know that lack or deficiency of MECP2 results Rett syndrome, but overexpression is associated with toxicity, as we see in MECP2 duplication syndrome. So to be able to rescue this MECP2 deficiency, it is very important to express this gene in a physiologically normal or a tolerant range. We believe that reactivation of X chromosome will not lead to overexpression of MECP2 for one, because we're using all the bells and whistles that are present on the endogenous gene. This is present within a cell. And second, the maximum increase that you can achieve is 100%. So we do not expect that reactivating X chromosome would lead to overexpression of MECP2 protein. Another important aspect of reactivating X chromosome is the expression of other X-linked genes beside MECP2. Data from my own lab, as well as previously published work from my pre-doctoral work and the postdoctoral work and other labs have demonstrated that activation of X chromosome, either by pharmacological means or by genetic means, do not lead to an increase in X-linked gene expression. Moreover, moreover, the expression of X-linked genes from the activated X chromosome could also benefit other X-linked diseases. For example, FMR1 for Fragile X syndrome and GDX3X for GDX3X syndrome. And there are other several X-linked diseases. So what we believe is that expression of MECP2, the healthy copy from the inactive X chromosome, can drive this defective neuron towards a healthy state, and which could enable to rescue the Rett-like neurological and behavioral deficit in the brain. So how do we achieve this? Well, my plan, to help you understand our approach towards this, I would like to use the analogy of a bulb in a room. If you want to turn a bulb on and off, you first have to figure out where the switch is. So that's exactly what my lab does. We use genetic screen, that is either using CRISPR-Cas9 guide RNAs or short hairpin RNAs, to identify the regulators of X chromosome inactivation. In other words, factors that keep the X chromosome in an inactive state. We use this information then to identify either small molecule inhibitors or other ways, other biologics, to turn this inactive X chromosome in a reactivated state. These genetic screens have enabled us to identify protein coding genes as well as non-coding RNAs like microRNA. Just to elaborate on what are protein coding genes, this is the flow of genetic information in human cells. We all know that DNA is the genetic storage of our information center. It gets transcribed into RNA, which is the messenger, which then gets translated into functional proteins that mediate all the functions in a cell. MicroRNAs are not the protein coding genes, so they do not transfer this and translate into a protein. Rather, that they go and bind to the messenger, the RNA, and leads to its degradation and thus regulate gene expression by down-regulating the messenger RNA. So how do we regulate these factors that we have identified through the genetic screens? We use two different approaches. For targeting the protein coding genes, we use a pharmacological approach, which means that we utilize chemically synthesized compounds to target the regulators, for example, activin receptor 1, PI3 kinase signaling pathway, or the Aurora kinase A. My lab has previously published and shown that these inhibitors, when injected into the brain of the mice, are capable of activating MECP2 gene in the brain of the mice. And the details can be found here. Another approach that we have utilized to target the non-coding RNA are the microRNA sponge. Now, microRNA sponge are synthetically engineered oligonucleotides that my lab has designed. And we use these sponges to basically facilitate the binding of microRNA, which is then lead to degradation of this microRNA and is depleted, therefore stops it from functioning in whichever pathway they are involved. In this case, they are involved in X chromosome inactivation. The next step after identification of these epigenetic regulators and their pharmacological inhibitors or the microRNA sponge is to test if this is actually feasible. Over the last few years, we have been using preclinical models to test these and rank them in the order of efficacy and efficiency to express MECP2. One of the models that we rely on are the mouse model. I would like to point out here that because this approach can only be used for female patients, we have been utilizing all the models, female models for our both cellular as well as murine models. One particular model that we are using here, which you are seeing is a mouse model that carries a green fluorescent protein reporter tagged to the MECP2 protein, which allows us to monitor the protein through immunofluorescence. Another modification in this animal is the deletion of one of the regulator of X chromosome inactivation, ZIST RNA, and therefore these females throughout the body inactivate only one X chromosome, which happens to carry the reporter gene. The advantage of this is that now none of these cells would express the green fluorescent protein. In other words, we will not see green signal. So if our biologics, if our inhibitors or our sponge are expressing the MECP2, we can measure and quantitate the amount based on the green signal. The data here shown is for a pharmacological inhibitor of activin receptor 1 and PI3 kinase signaling. And what we find is that there is a green signal indicative of MECP2 tagged to GFP reporter expression in the brain of the mice. Again, the details of this experiment and more experiments can be found in the reference listed here. In parallel to this, we also use another preclinical model, that is the induced pluripotent stem cell derived from the female Rett girl. This patient carries T158M mutation. And this model was generated by Alison Mittori and Fred Gage's lab, who were very kind to share this model with us. What you are looking here is the derivation of induced pluripotent stem cells and their translation to neural precursor cell and eventually neurons, which is the cell type most relevant to the Rett syndrome patients. What we did here is we monitored the response of neurons in terms of morphology and function to our drugs or microRNA sponge. And what you're looking here is just the top panel is for the neurons that are derived from the Rett patient. What I would like you to make note here that how cylinder they are, how thin they are, and how limited the extensions from these neurons are. These extensions allow the neurons to talk from one to each other and pass the message. When we treated our cells with our drug inhibitor, again here, the inhibitor is the activin receptor and PI3 kinase inhibitor, we see that these neurons are healthier, they have a bigger soma size, and a lot more extensions are coming out. We are doing similar experiments with all the other pharmacological inhibitors that we have identified, as well as our microRNA sponge. And those experiments are ongoing and the results are encouraging. So with this, I would pass on to my collaborator, Kathrin Meyer, who again, I would like to point out was introduced to me through Monica and Randy Carper. So I think this project was benefited from the facilitation that we got from Rett Syndrome Research Trust. But before I wrap up, I would just like to acknowledge my team who have played the very huge role in this project, especially Song Liu, who's the postdoc in the lab, and he is responsible for most of the data that has been generated for this project. I would also like to acknowledge our funding sources and especially the Hartwell Foundation who supported the early stage of this project, dealing specifically with microRNA. With that, I'll pass to Kat.
All right, thank you. All right, can you see my screen? And you can hear me, I guess? Okay. I'm very excited to be here today. Thank you, Monica, for the nice introduction. Indeed, I think we've met about 10 years ago, and I've been working on Rett syndrome for a long time now. And it was extremely exciting when you introduced me to Sanchita, because she's not only a great scientist, but I think in this process, we really also became friends. So it's always great to make new friends. So this is basically where I started my scientific journey at the University of Bern in Switzerland. I did my PhD in molecular and cellular biology. And I was actually already back then working on a gene therapy for spinal muscular atrophy. And that's how I got to meet Brian Casper. I decided to leave the mountains and the lakes and switch all out for the cornfields of Ohio which wasn't as attractive, but gave me much more time to actually work in the lab. So this is Nationwide Children's Hospital up here, and then Ohio State University. So the Center for Gene Therapy at Nationwide Children's Hospital was established by Dr. Phil Johnson and then really built up by Jerry Mendell, who's a real gene therapy pioneer. Our current director is Dr. Flanagan since 2017. And we focus on neuromuscular disorders and neurological disorders of children and adults. And to date, we have translated 13 AAV gene therapies to the clinic in the last 10 years. And AAV stands for adeno-associated virus, which you probably have heard of. And it's one of the major type of virus that we're using in the clinic at this point due to its outstanding safety capabilities. And there's also a couple of successful gene therapy startup companies that were founded by researchers of this institute. And, of course, this includes Avexis, of which you've already heard before, but there are others as well that are working towards treatments for kids. This is my lab, the Meyer Lab. We focus on neurodegenerative neurological disorders, as Monica said. And I was personally lucky enough to be already involved in 4 successful IND submissions, 2 different ones for SMA, intravenous, intrathecal delivery, and then 2 Batten disease clinical trials that are currently ongoing at Nationwide Children's. And we actually hope to get a 5th IND submitted this summer for another neuromuscular disease. So we're working really hard on that as well. But another aspect of my lab is also the development of in vitro modeling systems. And I'm also going to talk very shortly about that today. So as parents of Rett syndrome kids, I'm sure you're aware that the nervous system is really hard to treat because it's a really large organ. It has a lot of different cell types, but also it is protected by the blood-brain barrier. And that's a problem. And most of the drugs do not enter the brain very efficiently. And that's why we think gene therapy is really an interesting strategy for these kind of diseases. So what we usually do is we start our testing, as you heard from Sanchita, in the mice. But mice are pretty small. And also the mice that we have in the lab, they are very similar to each other. And in order to get to the clinic where we're really going to deal with very different individuals with very different genetic background, we oftentimes need large animal species to bridge that gap and understand the dosing, for example, for the clinic. But also one thing that's becoming more important now is that we can really learn from some of the clinical trials that we've already been involved in and really try to further adjust it, because even the large animals are different from humans. And so we can't always foresee everything that's going to be happening and how well something is going to work just from the animal studies. You probably heard about this. This is our first clinical trial that I was involved in, the intravenous delivery of SMN, the survival motor neuron protein, which is now FDA approved and is called Zolgensma. It was really a really incredible success. So based on that experience, what we find really is that the choice of the delivery routes and can really make a difference. So the production of these viral vectors is very expensive, and especially in the moment AAV therapy is booming, so the facilities are always booked, and then also the price is really, really high. So and in addition to that, the intravenous delivery for targeting of the nervous system also has potential to create some safety issues for older patients at high doses. And that's why we started really looking into the cerebrospinal fluid delivery. Can we take this virus and put it in the spinal fluid instead? And what we found with multiple programs, and this is just an example, when we did a side-by-side comparison with the intravenous delivery and the CSF delivery, that we can use up to 10 times lower doses and actually get the exact same effect, which you can see here. The green and the blue survival curve are exactly the same compared to the untreated animals here, but with 10 times less virus. One of the things that we have to consider also is that the amount of protein should be adjusted depending on the type of protein. So we can measure that by using reporter proteins. And here you see this green fluorescent protein, for example. So on the left side, you have a very strong promoter, which will give you a really bright green fluorescence. And on the right side, you have a promoter that gives you a lower expression level. So depending on the type of gene therapy that you're achieving, the type of protein that you're expressing, you want to choose one or the other. And this is particularly important for Rett syndrome, as Sanchita said, because MECP2 needs to be quite tightly regulated. So too much is not going to be good either. And as you know, we were involved in the development of the gene replacement program with Brian Casper that was also funded by the Rett Syndrome Research Trust. And here you can see Samantha Powers. She was a PhD student in my lab. She's now a postdoc, and she's still working with me on Rett syndrome and was already involved in this initial program. So this manuscript is still in preparation, but we hope to get it out still this year. And this is the approach that has been licensed to Avexis. We also use non-human primates quite often, just to, as I said, have a better idea on how the targeting will be in a larger animal that's more similar, but also to really do safety studies and see whether we have all our dots crossed before we go into the clinic. We don't usually do efficacy studies in non-human primates because those are very complex individuals. It's very expensive. We can't do as many non-human primates, and so the data is usually not as clear. So usually we use just healthy animals in that area. And this is an example using the AAV9, adeno-associated viral vector 9, which is the vector that we're using for most of the gene therapies at the moment in clinic that I was involved in. And what you can see is that you have a very efficient targeting of the nervous system and also the neurons. I'm showing you here just the spinal cord, but this is also true for the brain. And this is independent of the age of the non-human primate. So this is pretty exciting. However, here you see a distribution of green fluorescent protein measured with a different method throughout different regions of the brain and spinal cord. And one thing that I want to point out here, and that's why we ultimately decided to partner with Alcyone, and we're very excited about this, is that while we can target the entire brain with a lumbar intrathecal injection, the distribution throughout the brain is not even. So there are areas in the brain that will get more of your viral vector, and there are areas in the brain that will get less. And it really doesn't matter that much where you put the needle, whether you put it here, cisternal-magna injection, or lumbar intrathecal. It's very difficult to get an even distribution. And we are convinced that an even distribution will strongly increase not only the safety profile, but probably also the efficacy. So you get enough vector to all the areas in need, but you can avoid overexpression issues. Now, the way the vector spreads in your spinal fluid can be impacted by many different things. Of course, the dose and also the concentration of the vector, the formulation of the vector, so with what is the vector mixed, the injection volume, injection procedures, but also other factors that we either know or we don't know. Another thing that we need to pay attention to is that certain areas, especially the dorsal root ganglia that are next to the spinal cord, are very highly targeted with AAVs. And they can, an over-targeting of these organs can cause pathology. Although a recent study where Jim Wilson's group, who has actually initially described this pathology, has looked at all the data that is available from 33 different studies, and they did not find any clinical symptoms in 256 non-human primates that got therapeutic transgenes. So this seems to be a pathology that is fairly mild, but it's still something that we have to keep in mind and be careful about when we look at the dosing. And the delivery strategy will be absolutely key to make these kind of therapies more safe. As I said, in order to get an improved delivery, Sanchita and me decided to partner with Alcyone Therapeutics, and My colleague Bryn Martin will talk about this in much more detail. I hope you'll see why we're so excited about it. I think it really adds an additional level to the program that is going to make a big difference. But before we get to Dr. Martin, I wanted to quickly talk a little bit about the in vitro modeling as well. And so you probably heard about the technologies where we take patient skin cells, for example, and we can reprogram them either into pluripotent stem cells or directly in neurons or other cell types of the nervous system or intermediates. And these kind of reprogramming methods allow us to study the disease in a culture dish. And the Meyer Lab uses a shortcut of these reprogramming methods to make neuronal progenitors directly. And we study those, and we study neurons and glial cells. I'm going to show you a little bit of data from the Pitt Hopkins syndrome, which is very similar to Rett syndrome, as you know. But ultimately, using this method, we were able to identify a differentiation defect in patient cell lines that had a TCF4 deletion. So when you look at how they differentiate from the progenitor cells into the astrocytes, this is a marker for the progenitor cells, and this is a marker for astrocytes, you can appreciate that these cells were not actually able to properly differentiate. And so the Meyer Lab has spent the last couple of years to really develop tools to test our viral vectors in the tissue culture in more detail, because it would allow us to really look at different patient mutations and see who can respond to which treatment the best. But the AAV vectors are difficult to work with in culture. They work really well in the animals, but not so much in culture. But we were ultimately successful, and what we can see here is that the gene therapy vector that we developed was actually able to really push that differentiation and basically rescue that phenotype here. So we're super excited, and hopefully these in vitro methods, these methods within the tissue culture dish, will allow us to do a lot more testing also for Rett syndrome patients. So what you can see here, and again, this is the outstanding work of my close friend, Samantha Powers, who's been with me for many years now. She has developed a similar in vitro assay for Rett syndrome. And here you don't see the patient cell lines. They are underneath green fluorescent neurons. But what you can see is how the cells impact the neurons. And the different types of mutations have a different impact on the neurons. Some of them are more severe, some of them are milder, and some of them lead to an aberrant sprouting of the cells. So we have this assay developed that we now can use for testing as well. And another really good outcome from this study, and again, this was also supported by the Rett Syndrome Research Trust, is that we found potentially valuable biomarkers that we can use in our hopefully future clinical trials to really understand how the treatments are working as well. This was done in collaboration with Dr. Ferrariolo at the Sheffield Institute for Translational Neuroscience. With that, I would like to hand over to Bryn to talk a little bit more about the delivery methods that Alcyone is developing.
All right. Can you hear me okay?
Yep.
All right. I am going to shift over here. See if I can share. All right. Can you hear me?
Yes.
All right. Thank you, Kat and Sanchita. So I want to give a little quick introduction on the company Alcyone Therapeutics. I'm serving as the Vice President for Research, Precision Delivery, and CSF Sciences, which is a long term. My background is also as a faculty at a university with a faculty position in chemical and biological engineering, and I am still serving there as adjunct faculty. So it's great to be a part of the team here that really is looking into science and research related to Rett. At Alcyone Therapeutics, our aim is really to change the future of CNS therapeutics and to overcome challenges often seen in gene therapy by applying what we call precision CNS delivery, which I'm going to describe a little bit. We do have a pipeline, which you can learn about at our webpage. Among the pipeline is this ACTX Rett Syndrome therapy that we're working on. And so one key element that we believe is going to really make a difference in treating Rett syndrome is by applying precision CNS delivery platform. And we think that by really thinking about how we deliver the medication, not only what the medication is, but also how we do it, where we do it, and the way in which we do that, the dosing protocol and so forth, that we're going to have a better shot on goal to bring this therapy and have it be really restore health to the cells that Kat and Sanchita are talking about. This is a restorative therapy, the X-Reactivation. So it's really exciting to be doing this. We have a great team leadership. I joined in the last year, and we are really taking things forward from the lab all the way, hopefully very soon here, into the clinic. Okay, so when we say precision CNS delivery, what are we thinking ultimately? This slide encapsulates, I think, our grand vision, if you will. What we think we can do is we can actually take measurements on individual people with Rett syndrome. You see on the left-hand side here. So in this case, we're showing MRI measurements of the cerebrospinal fluid. And obviously, this is an important area to look at because this is where the medication is actually being delivered. So we need to understand that deeply, carefully in Rett syndrome, in that disease state, so that we understand better than anybody what's going on with drugs when they move in the CSF and how they move into the brain and so forth. So then what we can do is we can build engineering-based models that help us really optimize these therapies. And when we say optimize, we really mean bring together a very holistic, cohesive view of how to get the medication to the brain with the right therapeutic dose in the right way, right? With the right safety factors. And ultimately bring this solution even potentially to patient-specific cases. So, depending on your age, your sex, and so forth, we can, well, obviously in Rett, we have mainly female patients, but, we can take into account the details of individual people and dose them with the right system. So here's on the right-hand side showing application of our simple to use lumbar puncture based system. It goes in the lumbar spine here. And then we can dose people with a protocol that's going to get the drug to where it needs to go. All right, so now I just want to get into a little bit of detail. This is not meant to be comprehensive of how we're doing this. It's to kind of give the quick overview of the key elements. So in the top here, first thing we need to understand is what we call macro distribution of this medication through the cerebrospinal fluid. And the cerebrospinal fluid is not really very well understood. The physiology, how it changes in disease states and so forth. So I come from a background, essentially I've studied cerebrospinal fluid my whole life, have many publications in that area as a faculty, many different grants, work with industry, in foundations and so forth to really deeply look at cerebrospinal fluid as a diagnostic, also a way to get medications to the brain. That's the first component. Second component here is we need to understand microdistribution. How does that medication move along perivascular spaces into the spinal cord in the brain? You can see here. Then ultimately, we want to get this into the cells, which actually Kat and Sanchita talked quite a bit about. So my main focus is really going to be looking at micro distribution scale transport of this medication. Okay, so just zooming forward, we have a unique modeling capability. This is our human CSF system model. You can see here, it's a computational fluid dynamics model. It's really the world's most detailed, we believe, system for looking at cerebrospinal fluid transport. Has individual spinal cord nerve rootlets, radicular line, number of nerve rootlets, descending angle, all the details of the anatomy are integrated into this model, which we really believe is just a very elegant look of combined sort of medicine into engineering, where we can then use this model to simulate exact protocols, right? So here, what we're simulating, showing you, is a lumbar puncture delivery, which is sort of industry standard for delivering medications in this domain. And actually, I think my video is stuck here. Well, what you would see is as this medication is delivered, you would see it spread around the model. And I think the next slide also has some view of this. I apologize, for some reason the video's kind of stuck. So I'll show one more where you get a better idea for how that medication moves. But essentially here on this slide, you see in the top panel, the simulation result. So here is the human LP bolus, where it's a lumbar puncture, medication is delivered into the model. And the colored plot here is just a fancy plot showing where the drug moves over space and time. So on the x-axis here is time in hours. Y-axis is just position along the model, where we're showing that cross-sectional drug mass. And this is on a log scale. So log scale, if you remember from engineering class, it spans a huge range of values. So this is literally, tens of thousands, even millions in terms of the span. And what you see right away is that most of this drug actually remains in the lumbar spine. So this is a major problem. We do not want the drug all to remain in the lumbar spine. We really want to get things uniform, and especially around the brain. So this is our problem statement. And we've actually replicated these results in non-human primate models and found that in a similar way, we get very little drug to the brain when you do standard LP bolus protocols. We can quantify that in terms of area under the curve. This is an integral of this plot. And basically what this shows is that we're seeing more than 100-fold less drug to the brain than in the spine. So this is a real problem. We want to essentially flip this upside down. We want to get maximum drug to the brain, nice, uniform distribution, so that we really can have the therapeutic benefit of these drugs that Kat and Sanchita are bringing together. Okay, so here's another video kind of showing when we're plotting our results with our system compared to this standard lumbar puncture, which I showed on the last slide. And essentially what you're going to see is that our system can increase the dose to the brain dramatically. It can decrease off-target effects where you don't want the drug to just remain in the spine. And it also can thereby increase the number of people we can dose, right? We can get more to the brain. So effectively we have more drug available. So here you see this is a non-human primate model showing the intraventricular space, all the details of the model here. The nerve roots are similar to what we have in the human model. Again, this is very unique capability we have at Alcyone. And you see this side-by-side comparison of distribution when we use our technology versus a standard lumbar control, lumbar puncture control. So this is kind of giving the salient features. Obviously, there's a ton of details in exactly how this works. Okay, so bringing this forward is obviously what I showed so far was engineering modeling. And we know models can only go so far. So we need to actually test these results and see, is it really possible that we can see this kind of benefit using our system versus standard lumbar puncture? So here on this slide is showing our Alcyone precision delivery system where it's applied in an NHP model. You can see that this is gatobuterol imaging. You can see a huge amount of the medication, this gatobuterol, is delivered to the brain here on the top panel. Whereas in the lumbar puncture group, very little, almost none, is residing around the brain. We can also see this with PET imaging, positron emission tomography, where we can get a much better, more uniform distribution using our system compared to this manual LP. And this is, I should say, is, we're focusing also on this being a patient and therapy specific and also easy to use, safe system. So this is not doing something that's more invasive around the brain, like intracisterna magna or ventricular delivery. This is lumbar access for this system on the top. All right, so now zooming back out, I hope you can kind of see this vision that we need to comprehensively think about getting this medication to the brain. It's not enough to just look at the drug molecule on its own and the technology to deliver it. We need to also look at how we're getting it there, the protocol and so forth. And we think by looking at this carefully, we're really going to have a better shot on goal. All right, and that's all I had to present.
Great, thank you to all of you for really fabulous presentations. We have a number of questions, but I'm gonna kick off with a couple of questions of my own that I suspect other parents are gonna have as well. So when you showed the images of the MRI, and you said it's patient specific, how you would, I guess, set your equipment so that you're optimizing all the different things that can be optimized. Do you envision actually it will be different for every Rett patient, or are you envisioning coming up with a general set of criteria for your equipment that will apply to Rett syndrome versus other genetic disorders? Or is it really going to be person by person specific?
So to answer your question directly, is we do not know yet. However, we need full firepower to provide the right solution. So we do think we need to investigate all options on the table. Ultimately, we want the simplest solution. And we do think, there's going to be a range of what we need to do. But at this point, so I guess the quick answer is yes, we think that it can be more generalizable, but certainly you're going to need to have some parameters tuned in, for example, for age, things like that. So I don't know if that completely answers the question, but.
It does. And then my other question is the equipment that's attached to the catheter that physicians will be using. Is that something that they'll be trained on, or is that something that's going to require somebody that's not usually at a clinic or something to come in to do the procedure? Or is it simple enough that a neurologist can be trained on it and will be able to use the equipment?
So it would be simple to use, similar to an LP procedure that people can do, but it's still not without any risk whatsoever, right? I mean, even an LP has some risk, but we're targeting, but I would say, very simple to use.
Yeah, compared to like other options such as a cisterna magna injection or ICV injections, this will be a much lower risk. And for us, it was really important that it is a technology that can be used anywhere in the world. Like any hospital can do a lumbar intrathecal injection, and so it would be really something that is very easy to distribute throughout the world.
Okay. And I should say also is Alcyone has really deep knowledge in that area. Background on the company is that we do have a hydrocephalus shunt that has been brought forward previously, also intraparenchymal drug delivery in the brain. So we really have what I would describe as the full gamut of ability to do what we need to do. And, the experience needed to bring those kind of technologies forward, we're able to do that.
Okay, let's take some of our questions. We've got quite a few. We'll try to get through as many as we can. Okay, I'm going to take the first one from Tim Riley. If I understand correctly, this is for you, Sanchita. If I understand correctly, your results show that inhibition, elimination of exist is sufficient to reactivate genes on the X chromosome. Is this correct?
Thanks for your question. So just to reiterate, you're right that XIST is the key player of X chromosome inactivation. And there are reports and papers published that show that elimination of XIST can express X-linked genes. However, we are not completely eliminating cyst. What I like to call it as we are tripping the system to make it leaky, that is good enough to express X-linked genes, in this case, MECP2. So yes, your question is correct, but we are not completely eliminating cyst RNA from the system. I hope that helps.
All right, another question, this one's from Kathrin Romano. What is the duration of effect? Will repeated injections be needed?
I think Kat will take it.
Can you repeat it, Monica? Sorry, I didn't really
what
hear
is the what is the duration? Will it be a one and done, or will it be necessary to do?
Yeah. You know, it's a very good question. And I think we don't know it for any of the gene therapies. I mean, what we do know is that the therapy that we're planning here will be permanently expressed. So we'll have his own driver. So we are expecting this to be a one and done therapy. However, that we do know, and in neurons, the good part is that they're not dividing. So if you do gene therapy for an organ that is rapidly dividing, you lose the therapy over time, because these are not viruses that are integrating in your genome, because they can be dangerous. So we know from non-human primate studies that you can get more than 10 plus years, 15 years expression, but what we don't know is whether it's going to be 40 years or 50 years. So nobody knows. You know, these therapies haven't been in patients that long yet. So I think it's always good to also work on potential, backup strategies. But initially, yes, it will be a treatment that you don't have to take repeatedly, very similar to Zolgensma.
Okay, thank you, Kat. This is from Nick. What types of pathology can result with AAV treatment in the spinal cord?
Yeah, I mean, it's a good question. It always depends on the transgene. So what are you giving to the cells, and to which level as well? So you can have different kinds of pathologies. First of all, at higher doses, you can get pathology from immune reactions. You can get infiltration of immune cells into the spinal cord, which is not a big deal as long as it's moderate. If it comes to a place where the infiltration is not moderate, or if the cells start attacking, the cells where we gave the virus to, then it can be really problematic. Now, we haven't seen that in any of our therapies, just to say, but it is always something that could happen. The other thing that can happen is if you overexpress a protein and the cells do not appreciate it or cannot handle it, then it can also cause cell death. So you could get cell death in certain cases. But of course, before we go into the clinic, we do quite extensive safety studies. We are testing this not only in hundreds of mice, we're also testing this in larger animals, in non-human primates. And ultimately, if there is a doubt, we will not move forward into the clinic. Now, again, it's always a first in human, but we have a lot of experience, at least with AAV9 already. We already know a lot about the dosing with AAV9 and what levels are well tolerated. So that obviously helps us with the translation.
Okay, great. Here's a question from Beth Jones. Are there contraindications for intrathecal administration, for example, scoliosis surgery?
Yes, so scoliosis and surgery could be a problem, but I think Alcyone, again, is uniquely positioned to also treat children with complex spine conditions, because they do have the technology and the knowledge and the experience on how they can navigate through the spine, even though the spine has been injured or is deformed. Bryn, did you want to add anything here, or?
No, yes, I think that's correct.
There's a question from Laurent Villard. Thank you for the webinar. What do you think will happen in cells expressing the normal MECP2 copy when you reactivate the mutant?
Yeah.
Who wants to take that?
I can maybe start, and then Sanchita, you can. Yeah. So from what we see, really, and as a molecular biologist, is as long as you don't come in with a sledgehammer, which is the gene replacement is really the sledgehammer, because you come in with an external driver and a protein that only contains a small amount of the gene. A lot of the regulatory elements that we naturally have are not present in those gene replacement vectors. And so oftentimes, if the expression is natural from the natural endogenous gene, you don't get as high of an overexpression, because our cells actually have mechanisms to regulate expression by themselves, be it on the genomic level to just kind of slow down the transcription, as we say, or be it on the protein stability side and anything in between. Sanchita, did you want to add anything here?
No, I think you were on point.
Okay, there's a number of questions here about timeline. What is the timeline to get to a trial? And I know that's always a hard one to answer. But.
Yeah, it's very hard to answer, because we really want to respect all the families that are waiting for treatments. And I think the worst thing that we could do is give a timeline and then not be able to hold it. And especially now with what happened with COVID, that really has changed a lot of our timelines, for example, as well. But what I would say is that we're extremely committed. We're working aggressively on the preclinical studies, on the safety studies, and on all the aspects of vector production that we have to go through. And for us, this program is really a priority. So that's, we don't really want to give timelines, but we are very committed. We've been working on Rett for a long time, and we know that you all would really deserve to get some good therapies out there.
Thank you. And, I think there's peace of mind for me knowing that it's a lead program, because that means that a lot of attention is going to go to it. Yeah. You don't want to be number 12 on the pipeline. So in this case, we're the lead program. So that's good. There's also questions about patient age range. And that's probably too early for you to answer, right? And also, I know with the gene therapy programs that are in development now, it's always a big question about what the age range will be, and what will the FDA say about those age ranges. So the company could put forth a protocol with a particular age range, and the FDA could come back and say, no, we want you to go older, we want you to go younger. So I think until you've had those discussions with the FDA, and it's very difficult to say anything about age range, but I'm assuming you're going to want to have as broad an age range as possible, because that's good for the patient, that's good for the company as well.
Yeah, I mean, I think you summarized it really well. And it did happen several times in the past that because the gene therapies are considered a high risk strategy still, of course, that the FDA did require, at least in the beginning, to have patients that are a little bit further advanced, that are a little bit older, as first patients, first in human. However, the FDA has also seen that a lot of the therapies developed, at least for the degenerative diseases, have the best efficacy when applied early on. So it is really hard to predict. I think it will depend, on all the data that we are going to show them. And we have very intense discussions already now with clinicians to really work through these kind of strategies. But we haven't finalized anything yet. And even if we would, the FDA could still say no. Yeah.
Okay. Well, it's 1 o'clock, so we've run out of time. We'll try to, I'll try to address the questions that haven't been answered via email, and I'll probably be in contact with you with the panelists in order to make sure that we're answering appropriately. So thank you to our speakers. This was really phenomenal, and we hope that you'll come back and give us an update and inform us about your progress. And I think I speak for all parents when I wish you a lot of best wishes and luck and Godspeed, and work quickly. Thank you so much.
We will. Thank you very much.
Thank you.
Bye, thanks. Thank you.